Bismillah ar-Rahman ar-Rahim. We are going to talk about viruses and overview in this lecture titled General Virology. At the end of this lecture, you should know the general characteristics of viruses, including virus size and structure, classification of viruses, viral replication, laboratory diagnosis of viral diseases, viral cultivation and isolation, and finally, some important terms concerning viral infection. Viruses are the smallest infectious particles. Their size is measured in terms of nanometer. Their size is ranged from 19 to 300 nanometer in diameter. In comparison to other organisms like bacteria, viruses are much smaller. For example, E. coli is about 2 micrometer in diameter. So you can imagine how small the virus can be. The differences between viruses and other microbes. Viruses contain one type of nucleic acid, either DNA or RNA. Viruses are obligatory intracellular parasites. They can replicate only inside living tissue or living cells. They lack the metabolic activity and lack other enzymes and other constituents needed for independent growth and multiplication. They have a special replication cycle. Viruses are resistant to antibiotics and sensitive to interferons. Some viruses are able to induce latent infections by the integration of their genome with the genome of the host cell. The structure of viruses differs between different types, but as a general rule, all the viruses has a protein coat called viral capsid surrounding a nucleic acid core. Some viruses have other structures like outer lipid bilayer envelope. Viral capsid and symmetry. The capsid is a protein coat made of subunits called capsomeres. The arrangement of capsomeres gives the virus its structural symmetry. The geometric symmetry of the viruses are three types. Cubic symmetry, where the virus resembles a crystal called icosahedral. The second type is helical symmetry, where the virus resembles a ribbon. The third type is complex symmetry, where the virus has complicated structure. We have crystal-like structure, icosahedral, ribbon-like structure, and complex structure. Function of viral capsid is to protect the viral genome inside, also responsible for the attachment of the virus to specific host cells. Viral nucleic acid. Viruses contain either DNA or RNA. Most DNA viruses are double-stranded, except parvovirus is a single-stranded DNA virus. Most RNA viruses are single-stranded, except reovirus is double-stranded RNA virus. All the viruses have one copy of their genome, haploid, except retroviruses, which have two copies of their genome and called diploid. The nucleic acid is the infectious part of the virus, so colorless particles are non-infectious, as it carries the genetic information responsible for viral virulence and viral replication. Some viruses have viral lipid envelope. In general, the presence of such envelope confirms instability to the virus, as they are sensitive to heat, to drying and other detergents and solvents like alcohol and ethers. The term virion means complete infectious virus particle, nucleocapsid plus membrane envelope. Nucleocapsid means the viral capsid plus its nucleic acid. Viruses are classified into groups or families based on virion shape and structure, or type of nucleic acid, or method of replication. Viral replication. Steps of viral replication. First, attachment of the virus and adsorption to the host cell. Then, penetration of the virus and uncoating. After that, eclipse phase and synthesis of new viral components. And finally, assembly and release of new viruses from the cell. Again, the first step is the adsorption of the virus to the host cell. Then penetration of the virus inside the host cell. Uncoating and synthesis of new viral components, either nucleic acid or proteins. Assembly and finally release. Attachment of viruses and adsorption to the host cell requires specific binding between viral capsid proteins and a receptor on the host cell. For example, influenza virus have viral heme agglutinin, peplomer, that bind to specific cell receptor containing salic acid. HIV virus have a specific surface glycoprotein that binds to cellular CD4 receptor on T helper cells. This figure resembles a viral attachment to a specific receptor on host cell. After that, penetration of the virus inside the cell. The penetration is through formation of a vesicle and endocytosis in case of non-enveloped viruses. Non-enveloped viruses enter inside the cell through the formation of a vesicle that engulfs the virus inside the cells called endocytosis, while the enveloped viruses enter inside the cell through primary fusion of their envelope with the plasma membrane of the host cell. Other methods of penetration is direct penetration through direct injection of viral genome inside the cell, as in case of bacteriophage. Uncoating is a process in which the viral capsid is degraded inside the host cell, either by viral enzymes or host enzymes, and resulting in release of viral genome. Eclipse phase. In this period after penetration, there is no complete infectious virus particle can be detected inside the host cell, but the viral genome controls the cell to synthesize all the viral components, either nucleic acid or proteins. Virus replication involves synthesis of viral nucleic acid and synthesis of viral protein. The most important basic information you must remember is the central dogma and flow of genetic information through the transcription of DNA to messenger RNA. This messenger RNA is translated in the ribosome to protein. Again, the DNA is transcribed to messenger RNA. This messenger RNA translated in the ribosome to the protein. This is the normal central dogma. So the most important step in all viruses now is the formation of the messenger RNA for their replication. We have DNA viruses and RNA viruses, single-stranded and double-stranded, and the most important step again to obtain the messenger RNA or to transcribe their genome to messenger RNA that will be translated to the viral proteins. How to obtain this messenger RNA from DNA viruses and RNA viruses? In case of DNA viruses, the replication is occurred in the nucleus, as the DNA virus uses the host cell enzymes, which is called the DNA-dependent RNA polymerases, to synthesize their messenger RNA directly. DNA of the virus is transcribed to the messenger RNA. In case of voxovirus, there's some exceptions as the replication is occurred in the cytoplasm. In case of RNA viruses, the replication is undergo in the cytoplasm, except retroviruses and influenza viruses. We have 4 groups of RNA viruses according to their pathways to synthesize their messenger RNA. Single-stranded RNA viruses of positive sense or positive polarity, single-stranded RNA viruses of negative polarity, double-stranded RNA viruses, and retroviruses genome replication. In single-stranded RNA viruses of positive polarity, the viral RNA here is identical to the messenger RNA. That's meaning a positive sense or positive polarity. In case of single-stranded RNA of negative polarity, here's the virus required to transcribe their single-stranded RNA to its complementary positive sense RNA, which is acted directly as a messenger RNA. This is requires special enzyme in the virus called RNA-dependent RNA polymerases. RNA-dependent as it depends on the viral RNA, and RNA polymerase as it gives the RNA, messenger RNA. In double-stranded RNA viruses, which is a reovirus, it has its own polymerases to transcribe their messenger RNA. Retroviruses have its special replication cycle. Its genome is single-stranded RNA of positive polarity, but the virus here have unique replication cycle as it contains a unique enzyme called reverse transcriptase enzyme. Reverse transcriptase enzyme makes transcription in reverse direction. As we said before, the normal central dogma, DNA is transcribed to RNA. The DNA is normally transcribed to the RNA. But here in the retroviruses, the RNA of the virus is transcribed to DNA by unique enzymes called reverse transcriptase enzyme, as make transcription in reverse direction, RNA to DNA. This viral DNA is integrated to host cell DNA and then transcribed to give the messenger RNA of the virus, and then translation and give viral proteins. After replication of viral components, either nucleic acid and proteins, either early proteins, which are the enzymes responsible for the multiplication of nucleic acid, and late proteins, which are the structure of the virus, assembly of the virus, and release. In case of non-enveloped viruses, they are released from the cell through bursting of the cell or lysis of the cell and called cytolysis. But enveloped viruses typically released from the cell by budding. Diagnosis of viral infection. Diagnosis of viral infection either by direct examination through detection of the virus itself by electron microscope or some metabolites by light microscope, or detection of the antigen through immunofluorescence, ELISA, or other molecular diagnosis as PCR. Indirect examination through detection of antibodies by serological test. Any of the serological tests that studied before: viral neutralization test, heme agglutination inhibition test, complement fixation test, ELISA test. Direct examination by electron microscope, we detect the virus shape. By immunofluorescence, you can detect this fluorescence in positive cases. Light microscope to detect the viral inclusion bodies that are basically collection of replicating virus particles either inside the nucleus or inside the cytoplasm. Sometimes it is diagnostic, as in case of rabies. Intracytoplasmic inclusion bodies called Negri bodies found in the brain tissue is diagnostic. And molecular method, as we said, PCR or real-time PCR. Virus cultivation. Viruses are obligatory intracellular parasites, can't be cultivated on artificial media. It must be cultivated on living tissue or cells as eggs, cell culture, or animal. Cultivation of the viruses in embryonated hen's egg. This method cheap and easy to maintain and manipulated by the injection of the virus in the egg. The most popular form of injection in the chorioallantoic membrane, and if the virus is present, we will detect the POC lesion. The POC lesion is the white opaque areas on the transparent chorioallantoic membrane. That means the virus is present. Cell and tissue culture. Here, pieces of animal or human tissue are trypsinized to get a separate cells. These cells can grow in the presence of growth media containing serum in a special type of flasks or petri dishes, special type for tissue culture. This technique requires great precautions to avoid contamination, including working strike capping and sterilization of everything, gloves, the mask, and everything in surround. The tissue culture, after obtaining our cells, it can pass to other flasks. The number of this passage controls the type of tissue culture. Primary cell line, we can make several passages 4 to 6 times. This type of cells used for vaccine production. Secondary cell line, we can make passage for 20 times. And in continuous cell line, we use carcinoma cells, immortal cells that can divide spontaneously, as in case of HeLa cells from the carcinoma of the cervix. In this cell culture, we can detect the virus-induced cellular change that's called cytopathic effects. These cytopathic effects may be rounding of the cells, may be aggregation of the cells, loss of adherence, cell lysis, or fusion of number of cells to form giant cell or syncytia formation. Ballooning or rounding of the cells, or shrinkage of the cells, or syncytia formation. Lab animal inoculation can be also used. Some type of viruses that can't be propagated in vitro and in vaccine preparation also. And finally, the serodiagnosis is the most important step in the diagnosis of viral infection to detect the rising antibody titer. Now, some terms concerning viral infection are very important to be known. First, atypical virus-like agents. Defective viruses. Defective viruses are special type of viruses have a deletion of part of their genetic material. As a result of this deletion, they can't replicate alone. They need a helper virus to provide the missing function. For example, hepatitis D virus grow only in the presence of hepatitis B virus. Pseudovirus. This type of viruses, during replication, the virus takes part of the host cell genome instead of the viral genome, and so they can't replicate. Prions. Prions are infectious proteins. They contain no nucleic acid. Again, very important to know that prions are protein in nature, no nucleic acid. These proteins are encoded by cellular genes. These proteins normally present in the body as alpha helix configuration, but when they change their configuration to beta helix, they aggregate to form filaments which disrupt the neuronal function and result in irreversible fetal course in the CNS. These changes take long time, gradual onset, and long incubation period, so the prions diseases are called slow disease. It may take over 10 years. They are highly resistant to heat, UV light. However, they are inactivated by autoclaving. For example, of disease caused by prions is the bovine spongy encephalopathy in cows and Caro in man. Interference phenomenon. When two viruses infect one cell, this leads to inhibition of multiplication of one of these two viruses. Several mechanisms are involved. Number one virus may inhibit the absorption of the second virus by blocking its receptor, as in case of retrovirus and enterovirus. One virus may compete with the second virus for component of replication. Sometimes the first virus may cause the infected cell to produce large amount of interferon antiviral protein, as in case of Rubella virus and ecovirus. Interferon, it is the first line of defense against viral infection. It is the host-coded protein that inhibits viral replication and produced by the living cells in response to viral infection. Mechanism of action of interferon. It doesn't kill the virus. When the virus infects one cell, it stimulates the production of interferon by this cell. Interferon diffuses out of this cell to attach to the neighboring cell and stimulate them to induce antiviral protein called TIP protein or translation inhibition protein. This protein binds to the ribosome to prevent the synthesis of viral proteins. Again, when the virus infects one cell, it stimulates this cell to produce interferon. Interferon diffuses to the neighboring cell and stimulates the cells to produce very important protein called TIP protein. This TIP protein inhibits the viral protein translation. This is the mechanism of action of interferon. Now, what is the meaning of tropism? Tropism. Tropism is the affinity of the virus to certain organ or certain tissue where the virus multiplication occurs. What are the factors that determine the tropism? The presence of specific cell receptor or presence of specific metabolites or enzymes that activate the virus. Sometimes presence of some environmental factors as temperature is the cause of tropism. As in case of poxvirus, tend to locate in the skin because of their lower temperature. Persistent viral infection. If the virus escapes from the host defense mechanism, it gives either latent infection or chronic infection. Latent infection means that the virus remains hidden most of the time and for long period with intermittent reactivation. Intermittent reactivation and intermittent development of the disease course, as in case of herpes simplex virus. The virus is not detected during latency. It's only detected during the reactivation. But in case of chronic infection, the virus usually has long incubation period, but it always shed. That means we can detect the virus all the time. Example, hepatitis B virus. Oncogenic viruses means the tumor-causing viruses, resulting in transformation of infected cell. This occurs when the virus genome integrated in the host cell genome, as we said before in case of retroviruses. And finally, is there anything positive about viruses? Is there anything positive about viruses? Yes. Antivirus viruses are used successfully to deliver genes for gene therapy. Example, retroviruses. Also, some viruses can infect insects and so can be used as an alternative for chemical insecticides, which is called bioinsecticides. Example, nucleopolyhedrosis virus. Thank you and good luck.